हमने रैंडम प्राइमिंग, रेडियो लेबलिंग के हवाले से थ्योरी की बात की और उसकी रिक्वायरमेंट, रीएजेंट्स और बफर्स के हवाले से बात की। अभी हम बात करेंगे टू प्रिपेयर द प्रोब, व्हाट स्टेप्स आर इन्वॉल्व्ड एंड हाउ टू प्रोसीड फर्दर टू प्रिपेयर द रेडियो लेबल्ड प्रोब। सबसे पहला स्टेप इसमें requirement of template DNA and the primers. In a microfuge tube, add the required amount of DNA. Let's talk about 25 nanogram of DNA is added in 30 microliter of water. Volume is important because very high volumes cannot be handled later on. So 30 microliter should be the volume that contains 25 nanogram of DNA, and along with the 125 nanogram of the primers, the oligonucleotides, random oligonucleotides are added, and then after closing the tube we place it in the boiling water bath or that heating block at 95 degrees centigrade to incubate for 2 minutes. अब इस incubation period का जो 2 मिनट की incubation हमने रखी है उसका purpose क्या है? That is the denaturation time. In these 2 minutes, the double-stranded DNA is going to be denatured, and once the denaturation is complete, we shift this tube immediately to ice and we keep it there for 1 minute. So this shift in temperature from boiling temperature to the ice will not allow the 2 strands of the DNA to re-anneal or to renature. Instead, the primers present in the tube, they are going to bind to the template strand at their complementary positions. Once after this 1 minute incubation on ice, we centrifuge the tube. अब centrifugation का purpose क्या है? That we have very small volume at 30 microliter. When we place it at 95 degrees centigrade, the liquid portion is evaporated and is sticking with the cap of the plastic tube. So we have to get all the liquid component in the bottom of the tube. For that purpose we are going to centrifuge it, and that is going to happen at 10 seconds at 4 degrees centigrade. And after that we shift the tube back to the ice bath. The next step is addition of the components. We are going to add DNTP solution, 5 millimolar DNTP solution, 1 microliter each if we are using individual nucleotides, or if we are using the mixture of 3 nucleotides. Then we are going to add a random priming buffer, 10 microliters, and 5 microliter of the radio labeled probe, which is just arrived to your lab or to your facility. The final volume of this reaction mixture is adjusted to 50 microliters with the help of water. So once we add up the water up to 50 microliter, we proceed further for the addition of enzyme. After adding all the building blocks, all the volume adjustment, the next step is addition of Klinov enzyme. 1 microliter or 5 units are added and the mixing is done. अब mixing के लिए या तो हम vertex use कर सकते हैं, but vertex is not here recommended. We mix the enzyme with the 50 microliter of the reaction mixture by simple tapping on the tube like this. Using the finger we tap on the tube. Again the liquid is going to be disturbed and some of the liquid is on the walls of the tube or on the cap of the tube. We centrifuge the tube, all the enzyme, all the 50 microliter settles at the bottom of the tube, and now this is the incubation time. We incubate this tube containing our template DNA, containing our hexameric primers, then building blocks, DNTPs, radio labeled DATP or DCTP, and in the presence of Klinov fragment we let it proceed further, and incubation is done for 1 hour at room temperature. Once this incubation time is over, the next we have to stop the reaction, and for stopping the random prime reaction we use the stop or storage buffer. 10 microliter is added in the same tube, and then we have two choices to proceed further. Either we can store this radio label probe at -20 degrees centigrade for further use in the southern blot hybridization, or we can purify the radio labeled probe from the unincorporated radio labeled nucleotide using the spun column chromatography method. In this step, more than 50% of the DNTP or the radio labeled nucleotide is incorporated. We can see that the probe will have very high specific activity. यहाँ पे एक चीज़ का जिक्र करना बहुत ज़रूरी है. When we are working with radioactivity, we need to be very careful and we have to work behind the protective shields. We cannot handle the radioactive nucleotide or radioactive probe openly in the lab on the working bench. For preparation of the probe, there are defined areas where you can work with radioactivity, where there are available thick shields in between you and the working area, and you work behind a shield using your hands. Discarding the radioactive waste is also mandatory to be discarded in a specific container which is meant for radioactively labeled waste, whether this is a plastic tube, whether this is a pipette tip, or whether this is any liquid which is used in radioactivity.